Halo, Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
My student, ladies and gentlemen, welcome to our seminar under the scheme of 3-in-1, which is organized by Soil Science Department, one of the department at the Faculty of Agriculture, University of Brawijaya. It's been 3 o'clock in the afternoon. So in the next 1 hour, we will hear about the lecturers from our guests from Wageningen University, Professor Rachel Carmel. So just let me check whether Professor Rachel is here. Hello, Rachel?
Yes, I'm here.
Okay, great. So just let me introduce Professor Rachel Kramer is the head of soil biology group at the Wageningen University. So what time now at the Netherlands at the moment?
It's 10 a.m. in the morning.
10 a.m. in the morning, still in the morning, yeah, in the Netherlands. It's almost dark here, so 3 o'clock in the afternoon. Yeah, her interest is on the process on soil biology, range from the plot until a global scale. Is that right?
Yeah, yeah.
Yeah, previously, her interest is become a pedologist,
yes.
But after that, she is more concerned on improving soil quality and soil functions.
That's right.
How actually to manage soil and land to secure our sustainability of food production. Is that right?
Yes.
So as we know that the sustainable of the food productions become one of the major agenda in sustainable development goals at the moment. Yep. And it is because now there is also a raising concerns that the soil and the land is not just for maximizing the output of the food production, but also now become rely on its function and role on ecosystem, especially the land and the soil who provide actually nutrient for soil biology, and it become speed up the chemical process in the soil. So just want to say that Professor Rachel has published at least, at the moment I've checked in Scopus publications, at least 79 journal at the moment, and he has also become editor of Soil Use and Management Journal. Is that right?
Yeah, yeah.
Okay, I think in the next 30 minutes, time is yours, Professor Rachel Kramer, to deliver about your interest on soil biology, and we really want to know what the actual issue on this topics. So please, very welcome. Time is yours for the next 30 minutes. After you deliver your presentations, we will continue with question and answer. Okay, Professor Rachel, time is yours now.
Thank you very much. I just need to share my screen with you. Okay, can everybody see the presentation?
Yes.
Yes.
Yes,
yes.
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 Yeah, that's great.
Yes. Yes. Okay.
Yes, yes.
There we go. Okay, good morning, everyone. It's lovely to meet you all in a very virtual environment, and I really wish that I could be in Indonesia with you at this moment in time and meet you in person. But unfortunately, that's not possible at the moment. So here we are. This morning, I'm going to talk to you about the role of soil biodiversity in sustainable land management. And I'm going to, first of all, just touch upon what do we mean by sustainable land management. And then I'll introduce to you the what is soil biodiversity? What do we consider to be soil biodiversity? How does the food web work within the soil? And what does this mean in terms of some of the functions that take place in the soil? What are the biophysical characteristics of the soil and the soil habitat for the biota that's living there? And finally, what is the role of soil biota in supporting these soil functions that we are so reliant on as human beings in order to produce our food, but also to cycle our nutrients, to purify and regulate our water through our systems, and many, many other functions. If at any point you have questions, I'm sure that we can set up the chat at the side. If I can just see, maybe I can't see the chat, that's the only difficulty. Oh. So maybe feel free to raise your hand, and I will be asking questions throughout. So I'll also be looking for some interaction, and I hope to get some responses from you as well. Okay, so let's start. What do I mean by sustainable land management? Well, let's first of all visit the Sustainable Development Goals that were nicely introduced. And thank you very much for explaining that at the beginning of the lecture. I will presume that everybody knows something about the Sustainable Development Goals, but essentially there were 17 Sustainable Development Goals that were agreed upon in 2015 by all countries in order to ensure that we have a sustainable future. In order to ensure that really we have a fair and safe environment, and that we have food available to us, and that we're not damaging the environment. Now, for some of these sustainable development goals, soil plays a critical part. But I wonder which of these goals soil is really a main component of. And perhaps I could ask people to shout out which goals they think soil has a crucial role to play. Come on, we need some volunteers to speak up. Any ideas?
No, just go on.
There's 300 of you in the audience. You must have one idea of which of these goals soil may play an important part.
Yeah. Is there anybody want to select which part of the sustainable goal which is close to issue on soil and land management? So, anybody want to check on select which one is close to
Climate
the
action
on soil?
Sorry, speak up. Say it again.
Maybe climate action.
Climate action. Thank you. And I see Daly. Daly?
Yeah. Daily, yeah.
Yes. My question is the same with my friend before.
I need you to speak up for me, Daly.
Yeah.
I'm sorry if I don't sudah terdengar belum, Pak?
Maybe climate action.
Climate action. Thank you. And I see Daly. Daly?
Is climate action too?
Okay. Thank you, Daly. Okay, well, let's move forward a little bit then. So in fact, Daly, can I just ask you to mute your microphone again? Because I have, I can hear everything. In fact, soil contributes to actually 4 of the Sustainable Development Goals, that of zero hunger, good health and well-being, responsible consumption and production, and life on land. And interestingly, you would expect that soil would also contribute to climate action. But actually in 2015, soil was not mentioned as a key part of the Climate Action Sustainable Development Goal. And it's only since then that we've had discussions about how to deal with climate, that now soil has been put in as a crucial component of the climate action. So let's go through them a little bit. Interestingly, you would expect that soil would also contribute to climate action. But actually in 2015... I need everybody to mute their microphones if possible, please. And it's only since then that we've had discussions about how to deal with climate, that now soil has been put in as a crucial component of climate action. So let's go through them a little bit. Is it possible to mute your microphones? I need everybody to mute their microphones if possible, please. Okay. So if we look at sustainability goal 2, this is about ending hunger. So to ensure that everybody has access to food security and actually good nutrition. And the goal within this is to look at by 2030 that we have sustainable food production systems and resilient agricultural practices that support production and help to maintain ecosystems, so that we shouldn't be further damaging the environment through the production system. We also have Sustainable Development Goal 3, which is ensuring healthy lives and promote well-being at all ages. And this is really focused on pollution. And so what we don't want to do is, through agriculture, we don't want to further enhance pollutants within the water, for example, or within the air or soil itself that can cause disease or can cause ill health. Sustainable Development Goal number 12 is really looking at the sustainable consumption of our production patterns. So to ensure that we're recycling, where possible, waste materials and putting them back into the soil environment. So thinking about where we have manures from cattle, how these can be utilized in a safe way and reincorporated back into the soil. So the recycling of the system. And then we have Sustainable Development Goal number 15, which I think really is a critical one, which is about really life on land. It's about protecting and restoring all the habitats and the biodiversity that we consider both above and below ground, and to really halt further degradation. And I'm sure that this is something that every country in the world is experiencing at the moment, this extreme degradation of our soil systems, and then the associated habitats with that. And this can be through desertification, it can be through droughts, through flooding, but it can also be through many other forms of land degradation. And then, as I mentioned, finally, in 2020, not 2020, sorry, in the COP in Paris, where they brought together all the nations to talk about climate action, they decided that soil did indeed play a crucial role within the climate change strategies. And they started to implement within national policies, and I don't know whether this is the case within Indonesia, but in many of the national policies, particularly in Europe and across the world, there are now components where we look at the soil for carbon sequestration and for reduction of gas emissions. So to ensure that we don't further contribute to greenhouse gas emissions, and that we reduce climate problems, related problems. So all these sustainable development goals really rely upon sustainable plant growth. And sustainable plant growth is really about bringing together the various components of the agricultural production system in harmony, and to ensure that we provide good health from this for future generations, and that we protect the environment to ensure that future generations still have access to this land and are still able to grow nutritious food. And this really is about combining both above ground management practices that we think about in terms of primary productivity, the growing of crops. Here we have maize, we have some beans, also the production of animals in the system. So we've got cows in combination with the biota that lives under the ground, that also supports and enhances this primary productivity. So what the focus of the lecture will be about today. How can we do this? How can we make sure that our soil biota can really support our above ground sustainable plant growth? So now we talk about soil biodiversity. What is soil biodiversity? Okay. Well, here on the slide, you see a range of different soil biota, soil organisms that exist within the soil environment. Most of these are from temperate systems, but I'm sure that you have many, many soil organisms in the tropical systems as well. Does anyone want to tell me what they see here?
Yeah. Anybody want to tell what you see in the slide?
Worms.
Worms.
Worm.
Yeah.
Okay.
Worms.
Nematodes.
Nematodes.
Nematodes, very good.
Transparent one.
Centipede.
Microorganisms.
Centipede.
Yeah, which microorganism?
Which one?
And bacteria.
Bacteria.
Yeah.
Okay.
Very good.
Mm.
Okay, yeah, that's very good. That's a nice overview. So let's have a look at what we have here. So we have centipedes, millipedes. You can see the difference between the two there. Okay, so centipedes are carnivores. They will prey upon other soil biota, whereas millipedes are actually vegetarians. They're decomposers, so they eat leaf litter only. They're herbivores. We have bacteria and fungi, which really are the kind of the engine of the soil food web. And these really are involved in many, many of the processes that take place in soil. Isopods, so isopoda, which are fragmenters, they break down the leaf litter and contribute to the incorporation of it into the soil. We have potworms, which are otherwise known as enchytraeids. And these are actually a smaller version of the earthworm, but they look very different and they're much smaller. But in the right conditions, there is a huge biomass of these enchytraeids within the soil environment, particularly in acidic grassland or forest systems, which often take place at the role where earthworms are not present, because earthworms don't particularly like very acidic conditions, or many earthworm species don't like acidic soil conditions. So then the enchytraeids play a similar role. We have springtails, collembola, okay, and the mites. These are the mesofauna. So the enchytraeids, the springtails, and the mites really contribute as the mesofauna within the soil, and they're very much involved in nutrient cycling because they often graze upon the bacteria and fungi, as well as sometimes on plants, leaf litter, so on and so forth. And then we have the protozoa and the nematodes, which again are critical components because these will prey upon the bacteria and fungi. So this is only a small sampling of the kind of organisms that we find within the soil, but you can immediately start to see that they all have their own roles to play, and that they're all really important in ensuring how the soil environment works. And they're all different sizes. So actually our nematodes are from 10 to 100 micrometers. So very, very small. They're not really visible by the eye. You couldn't put them in your hand and see them well with your eye. You would struggle. So you need a microscope to look at those. Whereas the mesofauna, so that we have collembola, enchytraeids and mites here, are between about 0.1 and 2 millimeters in size. So the very small ones are quite difficult to see, but they're just about visible by the eye. And again, when we're looking at them in terms of trying to identify them, we would use microscopes to do this. And then finally, we have the macrofauna. And the macrofauna are between 2 millimeters and 20 millimeters in size. And I think actually if we go out to the tropics, probably even bigger. I've seen some centipedes out in Brazil, I have to say, which were enormous. And as a soil biologist, I wasn't ready to quickly jump into the pit and handle these centipedes. But again, these are the engineers. These are the movers of the soil materials, and we'll go on to talk about that later. So actually the size is really important in relation to the role that the biota have to play in the soil. And then in terms of abundance, well, generally, the smaller they are, the more number of individuals are present for a given meter square. So the microfauna, you can have up to 10,000 within a meter squared. Whereas the macrofauna, probably a maximum would be about 100 that you would find within a meter squared. And also in terms of the number of species. So the macrofauna, you may only get 10 different species found within that 1 meter squared. But again, with the mesofauna, you get possibly up to 100 different species. And for the microfauna, again, about 100 different species. So the soil is incredibly diverse and incredibly highly populated. It's quite amazing how much lives beneath our feet. And then we come to the bacteria and the microflora. And again, then we're talking about 10,000 up to 1 million individual organisms within a meter squared. So we've thought about now how we, what we have in the soils, but how do we also think about what they do within the soils? Well, we do that through different forms of classification. And so we can look at the taxonomy, so we can describe them, we can speciate them. And in many cases, this is done for the macrofauna by visually, by the eye. We can describe what we see. Earthworms, for example, we can count the number of segments, and that enables us to come up with different earthworm species in combination with the colour. We can also use microscope for the mesofauna and the microfauna. And really, once you get to the microfauna level, you really need specialists, possibly even with the mesofauna. I remember within my PhD, when I was studying, I would look at collembola and mites and nematodes and enchytrates in the soil. But to become a specialist in all those topics was incredibly difficult. So actually what I would do is I would try and organize the different individuals that I would collect from the fields down to a level that I was able to do it. And then I would send each of those batches off to further specialists who would then speciate it down to the species or genera level. But we can also think about classifying them according to body size. So I just explained to you that actually the body size can be very important in how we distinguish between these different groups and the roles they have to play. Another way is to think about trophic groups. So where do they live within the soil? What kind of food do they eat? So if we think about nematodes, and I'll go on to describe this a bit later within the food web, there are different types of feeding habits that those nematodes could play. So some of them are parasitic, some of them are predatory, some of them are omnivorous, some are herbivores. And so depending on what they eat, also changes the role that they play within the soil. So we can classify on that basis. And finally, we can think about functional groups. And this can often be how they contribute to specific processes that take place in the soil and their role within that. And then when we measure biodiversity, I've talked about how we can use microscopes to study them, but there's other ways to study soil biodiversity too. So if we think about the bacteria and the fungi, actually most of the time now we would use genotypic descriptions of the soil microorganisms. And by this I mean using DNA. So this could be different forms of DNA extractions that we do, whether we want to just look at the bacteria community or whether we'd also want to look at the fungal community. And we can also think about very new methods where we apply things like metabolomics or proteomics to look at some of the components of the gene pattern, the blueprint that we find within the microorganisms. We can also look at the phenotypic, so the expression of certain parts. And here a good example is the phospholipid fatty acid methodology, where actually every cell wall has a particularly unique identifier for different species. And by doing that, we can examine, we can extract that part of the cell wall, the phospholipid, and we can then run it through and we can determine whether we have gram-positive bacteria, gram-negative bacteria. We can determine fungi that's present in the soil, so on and so forth. So there's different ways of looking at the microbial community. And then finally, we can also look at the functional relationship of the microbial community. And this is where we can actually run assays within the lab. So we can actually measure some of the processes taking place and see who is responsible for doing that. Okay, so I've explained who we find in the soil and how we would measure those different components in the soil. And now I want to think about the role that they play. So for food web interactions, by this, I mean who eats who in the simplest of terms, because actually the soil environment is of course made up of all these different organisms of all these different sizes who all have different feeding habits. And we need to be able to understand the relationship between who is the predator or the prey in these different systems. And so what you can see here is you have your tree with your roots and your mycorrhiza, which can express drop leaf litter, for example, onto the ground or produce root exudates. The mycorrhiza also looks for nutrients, scavenges nutrients in association with the tree. And that in combination then with fungi and the leaf litter decomposition starts the process of decomposition. And this is then where we have the dung beetles, the millipedes, the isopods, the earthworms and the enchytraeids, or the potworms that come in, and the mites and the nematodes who start to break down that leaf litter material, or in the case actually may scavenge directly onto the rooting system of the plant itself. And they start this initial process of the breakdown of organic matter that comes into the system. So leaf litter material, okay? Now, this can be, here I've given an example of a tree dropping leaves, but it can also be material that's left in the field following a harvest. And these animals not only fragment it, they break it down, but they also incorporate it into the top. And so we have, following that, we have animals that further break it down again and contribute to the humification of this organic matter in soil. Of course, we also have some top predators. And these top predators can eat upon these macrofauna as well. So let's look at this schema from a slightly different perspective. And now we can look at the carbon flows within the soil. Because actually, what all these organisms are doing is they're looking for energy. And the energy is obtained through the carbon that's present in the leaf litter material or the root exudates that's coming into the soil. And so when we fragment or break down the material, the macro fauna, for example, they're consuming it in order to obtain the carbon in that material to give them energy. And that energy flow goes all the way through the soil. And so the carbon and the nitrogen that's present in the material that's deposited on the soil flows all the way through this food web system. And that carbon can be captured inside the organisms themselves, because of course, when we eat something, when we eat some food, some of it is retained within us, okay? But some of it passes through us, and of course we excrete it out, and then another organism may come along and break down that material that's been excreted out by some of these larger organisms. But some is always retained within us. And this is constantly cycling through the soil system. And so actually what the food web does is, while it's about who eats who within the soil, it's also about the flow of carbon and nutrients throughout the soil system. And this flow of carbon and nutrients isn't just good for the soil organisms, the soil biota, is also good for the plants because it helps to release the nutrients, which then provide a form of fertilization to the plants through the organic breakdown of litter material. So when we think about the flow of carbon and nitrogen in soils, there's three major processes at play. As our leaf litter drops onto the surface of the soil, we have the fragmentation of this by the macrofauna and some of the mesofauna that chew it up, retain some of it, excrete some of it. Then the microbes come into that, and the bacteria and the fungi, they move in onto these excreted products. And actually, in the case of the fungi, they can also do it directly onto the decomposing litter material. And they degrade, they further degrade and break down this material that's entered the system. And then those bacteria and fungi are then themselves eaten by organisms that are higher up the food web. So by nematodes, by enchytraeids, by mites, by collembola, so on and so forth. And the cycle continues. And this whole process is an incredibly sophisticated process of nature. So nature in itself completely ensures this turnaround of carbon and nitrogen within the system. And what you can see here in the diagram is we've got the decomposition that takes place with the leaf litter that's been dropped onto the soil. That goes into the biomass carbon. And what I mean by biomass carbon is taken into the bodies of the organisms themselves. And when those organisms themselves excrete or either die, which often in the soil environment, the turnover of organisms is very high. They have very short lifespans, some of them. The larger ones have larger lifespans, but the microbes have a very fast turnover. Then that material is further humified. So it's actually stabilized within the soil system. And it can go through the cycle of the biomass many times, and small parts of it are then further stabilized within the soil organic matter. And when I talk about stabilized, I mean that they're actually incorporated into physical parts of the soil material or chemically bound with the soil material. And so they're held for a long time. And of course, throughout this process, the soil organisms also respire. And so they produce CO2 because when they're consuming this organic material, they're producing CO2 as a result of it. And so there is a constant flow or loss, if you want to think of it, of CO2 from this system. So by adding organic materials into our soil, it's very good for our soil. Our soil actually needs, carbon provides, as I say, the food for all these organisms to not only live, but also to do their jobs within the soil, which are critical for us and for the functionality. But of course, that also incurs respiration. So whenever we add more, we respire more. It's a cyclical process. But where you can see now that we have a loss through the respiration with the CO2, we also gain, because we gain the carbon that's within the biomass, and we gain the carbon that's then stored and stabilized in the soil organic matter. And this stabilization can be short-term, medium-term, and even long-term when it's associated with the silt and clay fractions in the soil. And so we, over time, can store a lot of carbon within this system. So just to sort of recap the major players that we have here of the fragmentation, the breakdown of the organic material are the earthworms, the isopods, the mites. And then for the degradation, it's really about bacteria and the fungi. And this is managed through a process of grazing by the collembola, the nematodes, protozoa and mites. Okay. Is everyone still with me? I hope so. Okay. So then I thought we'd just touch briefly upon nematodes, because nematodes are ubiquitous. They're found everywhere. They're in every living environment in the soil across the globe. And so they play a very critical role within the soil because they're grazers of bacteria and fungi. And as such, they enhance nutrient availability in the soil. So by grazing upon the bacteria and fungi, they're releasing the nutrients that are held within the biomass of bacteria and fungi. It can also be plant and animal parasites. And so sometimes we find that we have a lot of nematodes that can cause huge amounts of destruction and devastation within the agricultural cropping system. So we have to be careful that we don't allow one particular group of nematodes to build up within the soil. But by having a very diverse population of nematodes of many different roles, then we can manage that. And they also are prey themselves for meso and macrofauna. So they provide a food resource and the energy associated with that to the meso and macrofauna. They can be primary consumers through feeding on plant roots and the plants themselves. They can be secondary consumers with the bacterial and fungi feeding. And then they can be tertiary consumers because we also have predatory nematodes and omnivorous species. And omnivorous means that they have no specific feeding habit. They will change their feeding habit depending on the environment that they're in.
Oh,
And they can play, yep.
Would you please to show the previous slide before the last one, which is before this one? Yeah, this one. So yeah. According to this process, we can have a look at which one the process is actually coming first, which is we would like to focus on fragmentations of the dead material should be coming first, or we should prefer the role of bacteria and fungi as the microbial degradation. So we end up with the more smaller particle instead of the bigger one, which is degraded by fragmented by earthworm or other bigger macrofauna in soil. Which one is actually should coming first?
Well, I think it's a cyclical process, in fact. So the bacteria and the fungi can already be degrading the leaf litter material, for example, but it's very hard for them to completely consume that material on their own. So the fragmenters then come in and help break down that material. So they work in combination. It isn't necessary that one has to take place before the other. The two work alongside each other.
Okay. Thank you.
No problem. And so I thought we'd briefly touch upon the nitrogen cycle because I've just talked about the flow of carbon in soils, but with the flow of carbon in soils, of course, we also have the flow of nitrogen in soils. And nitrogen is a very, very important nutrient for the growth of our plants in agricultural production systems. And actually the soil biota play a critical role here. And we have specialists within the soil that help in the process of the nitrogen cycle. So we have rhizobia, for example, which are nitrogen fixing, okay? So these work, they nodulate specific plant root systems, and they can actually capture nitrogen themselves. So they fix the nitrogen. We also then have nitrifying and denitrifying bacteria. So these help in the process of the conversion of nitrogen from ammonia in the soil to nitrate through nitrification. And then further in anaerobic conditions, they can then denitrify the soil into N2. And if it's only partial denitrification, N2O, which of course can be a greenhouse gas. So we want to have the best conditions possible for denitrification to ensure that it goes back to N2. So they really have a critical role to play in the mineralization of the nitrogen that takes place. And they do this in combination with trying to consume the carbon that's available, because the carbon and nitrogen are linked together within the leaf litter that's being broken down. So I want to move away then from the chemical cycles that are in the soil.
Rachel?
Yeah.
It's already some questions coming into the chat room.
Okay, I can't see.
And then we would like to read one by one, and then maybe if you have a time, you can answer this question first before you continue with the next slide. So we will check the questions that's already inside the chat room. So, yeah, this is the question from Riandra. So actually, for our students, it's not necessary asking in English. You can also ask in bahasa ya. Mohon maaf, in bahasa tidak apa-apa. We will transfer the bahasa into English. Don't worry. So please raise your questions, and we will have to transfer it into English. Ya, bagi mahasiswa yang memiliki pertanyaan, silahkan ditulis di chat room, akan kita pilih, dan kalau memang di bahasa Indonesia tidak apa-apa, nanti kita transfer ke bahasa Inggris. Kami persilahkan untuk menulis di chat room. So yeah, the first questions actually relate the problem with the soil toxicity and the soil quality. So which one is the most critical one? Should we focusing on the degradations on the soil in terms of the raising of the soil toxicity, or we should improve the soil quality? Which one is the most important at the moment, Rachel?
Well, that depends upon the condition of the soil. So obviously, if you have an incredibly toxic soil, the quality of the soil is very bad. But I think toxicity is a major issue within soils, and contamination in general is a major issue. But there's no point in only focusing on one form of contamination in the soil or degradation in the soil. You've really got to think about actually how we can ensure that that soil is able to be a good quality and actually support all the biodiversity that's in the soil, and support the processes that soil needs to do in order for us to have good plant growth and good environmental conditions. So yes, toxicity is a massive issue. And I think that in many countries, and perhaps Indonesia is one of those countries, it's a really big problem. But there are other degradation issues as well that need to be considered.
Oh yeah, thank you. And our student is also being confused. At the same time, we're also facing a problem with the global climate crisis. So which one is the most important thing to handle, yeah? So we're facing with the soil degradation of the soils, soil quality also need to be improved, and the climate crisis is also on the way. So they also a bit.
So the soil does have a large role to play within the climate crisis, but it doesn't have the only role to play. So I think when we think about climate, we also have problems with industry, with the transport sector. There's many other issues contributing to the climate crisis that we really need to pay attention to. And actually, good soil quality can really then support further reduction of emissions from the soil, which would then contribute to the climate issue. So it's all about trying to ensure that we have good soil quality overall. Then we can cope with many of these things.
So yeah, you mean that we have to focus on improving soil quality. By that way, we also contribute on reducing the climate issue then.
Yeah.
Okay. So we continue with the next questions, actually focusing on how actually we could manage the soil within this period. It is because we could not guarantee the optimum conditions of water or air in the soil, which is really need by soil microorganisms to grow, to be active, and play the role on decomposing soil organic matter. It is because the climate has been changed, the rainfall has been reduced. We could not guarantee that the soil in the optimum condition for habitat for soil microorganism. How you can give your idea how actually this process?
Yeah, this is a big problem. So actually, dealing with the soil needs a certain level of moisture in order for the organisms that live within it to survive. If it's completely dry and you have severe drought conditions, the plants and the organisms within the soil will deteriorate. They will die. So you need to ensure that we do the right management practices to ensure that when we do have rain that comes into the soil, that water is held for as long as possible. So actually it's really about, and again, carbon is a critical part here. If we take the leftover residues, for example, from a crop system, and we incorporate those back into the soil and the organisms are breaking that down, this provides a good soil structure. And the soil structure is critical for holding on to that water. If you have a very poor soil structure in the soil, then you very quickly lose any water that's put into that system. And so actually the organisms play, and I will talk about that in the physical habitat now, a critical role in not only looking after the structure of the soil, but also the conditions that structure then supports, such as good air capacity, good water holding capacity, and they then protect that soil in the longer term. So when you have then a drought period, if your soil has good water holding capacity, it actually can retain water for longer and support the organisms and the plants for longer. Obviously, there comes a point where it becomes extreme in a drought situation, and we then hit something called the wilting point, which means that there really isn't enough water available in the soil to support the animals and plants.
Okay. There's still several questions, but it might be related with the slide on how actually the role of nitrogen fixing bacteria in the relationship on restoring soil quality.
Okay. Well, I suppose it's in relation to how we fertilize the soils. So the presence of nitrogen fixing bacteria within the soil will enable natural fixation of nitrogen in the soil. So rhizobia, or if we grow legumes, for example, they contribute the natural nitrogen from the atmosphere into the soil. Now, often this isn't enough to then supply and support crops as we grow them within our cropping systems, within our primary productivity. So then you have to decide, how will I support that natural fixation with the management of the fertilizers that I apply into this system? And we can think about inorganic fertilization. So this is fertilizers, inorganic fertilizers that we apply in the form of nitrate that goes straight to the plants. Or you can do it in an organic form. So you can also think about applying, for example, organic manures, so from the animals, back into the soil, and that in itself contains nitrogen, and that can also support. often we use fertilizers because they provide a faster response by the plants. So the plants respond much quicker when we apply inorganic fertilizers into this system. But it's not necessarily always the best option, or we often can over-fertilize by adding too much inorganic nitrogen, which then has really bad effects upon the environment around it. If we have too much nitrogen in the soil, we get nitrate leaching, which contributes to eutrophication. Or we can get the production of N2O in very wet conditions, and that's obviously a greenhouse gas. So it's managing how much nitrogen is in the soil. Rhizobia helps that, but probably on its own isn't totally sufficient.
Okay, yeah. So one more question. So is that correct that the first step for maintaining soil the abundance of the soil microorganisms in soil is just providing organic material? Is that correct? Or, okay.
So it's food. Microorganisms also need food to survive. So there needs to be a form of organic material in the soil for those microorganisms, and also for many of the micro, meso, and macrofauna within the soil.
Oh yeah, one more. There's a lot of list of questions. This is from Prof. Cho. Maybe you know her, Rachel Kurniatun Arya.
Possibly, yeah.
Yeah, possibly, yeah.
I'd need to.
It's about the questions about more. Some farmer assumes that the earthworm is harmful for coffee root system because they assumes that sometimes they're eating this kind of root. I don't know exactly whether the root is one of the earthworm preferences. So do you have any such experience about this, that some earthworm usually also prefer eating specific root which might be suitable for survive?
So I think this is probably quite specific to the climatic conditions that you have there in Indonesia. I can't tell you of any problems that we have with earthworms in temperate systems for eating specific rooting systems. Yeah. We certainly do have problems with nematodes, which often are plant root eaters. So they will destroy a lot of plants, but not earthworms as such. So I would need to know more about what species it is, and I'd have to look it up.
Okay, yeah. Your opinions maybe not just because of the fact of the earthworms, that might be nematode contribute to this process, yeah?
Well, I think I would need to know more about the earthworm in particular, because maybe it's something very specific to the climatic conditions in Indonesia that we don't experience in Europe.
Okay, so still several questions, but we will wait until your presentation completely finished. Please, back to you.
Okay, so now I'm going to talk about the physical environment or the physical habitat within the soil and how that is developed. There we go. So we've just been talking about earthworms and obviously farmers worrying about particular earthworm species within their environment, but actually earthworms are incredibly important within the soil system, and they're also incredibly vulnerable. So we talked about acidification and contaminations, and because earthworms are soft-bodied animals, they often are hurt or killed through contaminants and through acidification. So we really need to look after our soils in order to look after our earthworms, because they play a fantastic role. So their main kind of characteristics is that the earthworms can help with fragmenting the leaf litter material that we talked about in terms of decomposition and the carbon flow and nitrogen flow through the soil. So they really have, and they consume it, and then they produce earthworm casts, and this is full of nutrients and carbon that the bacteria and the fungi then work upon. They also Anesic earthworms, so this is a particular type of earthworm, and I'll explain it in a minute. They pull the material, the leaf litter material, down into the soil itself through their burrow system. So there are very specific species that come up to the surface and pull the material back down. And then they incorporate it further down within the soil profile. And then the other role that earthworms play is they bioturbate the soil, which means that they mix it. They mix the mineral particles, the physical particles within the soil, the sand, silt, clay, the textural units, with organic matter. And these produce the structural units that we describe as soil structure within the soil. And these are critical in terms of the porosity of the soil, in terms of the water regulation through the soil, so the movement of and storage of water in the soil, and also in terms of the amount of carbon material that's available for all the other organisms within the soil. So earthworms play this incredibly important role. So we shouldn't fear them. We should really, I think in the majority of cases, we should really look after them. And as I said, there are 3 different types of functional groups of earthworms. There are the epigenic earthworms, and these live literally in the top 2 centimeters of the soil. And they just feed on that plant litter material. Then we have the anesic ones, and these were the ones that I described. You may often see that in the morning, the birds are in the field and they're pecking into the soil very early in the morning. When the earthworms come up to the surface, they grab material from the surface and they pull it back down. And if a bird is fast enough, just as the earthworm comes out of its burrow, the bird can catch it. But earthworms are very clever. And actually what they have, these anesic earthworms have a special anchor on their tail, which actually they keep within the top of the burrow, and they search for the leaf litter material, and then they go back down into the burrow again. And sometimes when you see the bird pulling at the earthworm, it's because the earthworm's tightly got its tail still within the soil. And then finally is the endogenic earthworms. And these earthworms live all the time deeper in the soil. So these will live around the 10 centimeters to 20, 30, 40 centimeters depth. And they burrow through the soil. And as they burrow through, they consume the soil and then they excrete it out. And this actually really adds to the mixing and bioturbation. And I got this very nice little cartoon from Felicity Crotty in the UK at the Royal Agricultural University, where she shows that actually there's a greater biomass or weight of organisms below the ground in earthworms than are grazing above the ground. And you can have up to 3 tons per hectare of earthworm biomass under the soil. So that's quite incredible, because we look at a cow, for example, and we think, well, that weighs a lot. But we have that as well underground through the earthworm populations or the Enchytraea populations. And so they really do play a critical role. So what is bioturbation? Well, bioturbation is this major physical movement of material, be it the biotic, the carbon or associated biotic products from the organisms, or the abiotics, so the physical parts of the soil, the sand, the silt, the clay, and these together form soil structure. And they also create micro and macro habitats for the earthworms to live in, but also for all the other organisms to live within. And they can modify the availability of the resources. So as I say, they can incorporate organic material further down into the soil profile itself. And if I can I just have to see if I can switch out of this screen and I will show you, if it's possible. Let's go to it. I have the internet here. Let's try this. This is a video that was created by my group here at Wageningen University. And this really shows the roles of earthworms. So what we have here is we have on the right-hand side, where you can see all the activity taking place, we have earthworms present within this column. with the leaf litter material and the soil underneath. And on the left-hand side, we don't have any earthworms present. And what you can see is taking place is that the earthworms are breaking down that leaf litter material that is on the surface of the soil, and they're slowly incorporating it into the mineral soil underneath. So the sort of darker soil, and you can actually see that the earthworms are starting to make these burrow structures as they decompose this litter material. And it's absolutely incredible that over a matter of a few weeks, these earthworms can completely break down all this litter material and incorporate it into the soil. And as they do that, of course, all the other soil organisms start to work upon this and break it down further. And I'll just stop it there. We'll switch back to the presentation. Okay. So earthworms play this very critical role, but it's not just earthworms, as I said. Where there is a lack of earthworms, then we often find enchytraeids in these environments. So these are the potworms. They're from the same order as the earthworms, but they are white worms, and they tend to live in more acidic conditions. Ants and termites are also major ecosystem engineers or soil engineers, whereby they contribute to the breakdown and bioturbation, breakdown of leaf litter material and bioturbation, so incorporation of this and the movement of the soil particles. So as I mentioned earlier, we have these 3 different types, and I just give you some examples here of these different types. We have the Epigenics, which are often very colourful in colour because they're near to the surface, so they tend to be more colourful because they're in the sunlight. We have the Endogenic, so the soil dwelling, and these are very pale in colour. They never receive any form of sunlight. They're always permanently within the soil burrow, so they're very pale in colour. And then we have finally the Anesics, the leaf litter feeders, which have these permanent burrow structures, and you can see it here in the diagram, where they have these burrow structures that go up and down, and they will have a few burrows that they utilize on a regular basis. And what happens is that the upper part of the body, the reddish part of the head part of the body tends to be slightly more pigmented, and then the tail part is often less pigmented. So you can see a color differentiation across them. So we use color as one of the ways to classify the earthworms, in combination with counting the number of segments that we find on an earthworm and the saddle where the reproductive organism place. So we talked about bioturbation. Well, what are they actually doing? They're physically protecting, so they produce binding agents that bring together the physical particles, the sand, silt, clay in the soil, and make those into aggregates. They contribute to that. This can also be done by roots and fungal hyphae as well. And then this can bind further with the help of bacterial gums that really aggregates this material together. And this can then also hold this carbon onto the soil in the longer term. So it also contributes to carbon storage. So finally, I talked a lot about who lives in the soil, what they do in the soil, and who eats who in terms of the food web, and the flows of carbon and nitrogen, the ecosystem engineers and how they really contribute to the development of the soil structure and how important that is in terms of the moisture conditions and the temperature within the soil. And finally, I want to talk about soil functionality. So at the beginning of this presentation, I described to you that soil biota were really critical in terms of sustainable plant growth. And I showed you in that picture a mostly above ground image of the crops that were being grown and of the animals grazing with a small section of the soil. Well, now we move it down and we look a little bit more at the soil and of the interactive role that the soil biota plays in supporting that sustainable plant growth. And so they do this through nutrient cycling, through the management of carbon, be it carbon for the organisms within the soil, carbon in terms of helping to release nutrients, therefore contributing to plant productivity, but also carbon sequestration, so long-term storage within the soil, they contribute there too. Disease suppression, so actually by having a really diverse range of biota within the soil, across all these different groups that I've described to you today, it can also contribute to suppressing any diseases developing in the soil. And then finally, water regulation and purification. So the water that, when we have rain that drops on our soil, it infiltrates into the soil environment and it goes through the soil matrix. And it does that by the macro channels that are produced by the roots and the earthworms, or the ants or the termites in the soil, but also it's purified as it moves through the soil. So it actually interacts with the mineral particles, with the bacteria, with the fungi, and often contaminants are sequestered out of the system, and that water eventually moves into the aquifers deep in the rock system below, or into the stream system through flow of the soil down a slope. So the soil plays an incredibly important role in looking after not only our agricultural systems, but also looking after us in terms of ensuring that we have all the needs that we have from the land is supported by the organisms and the soil itself that we grow these plants on. So I gave a few key examples here for each of these soil functions. So for carbon sequestration and climate regulation, probably one of the main soil biota responsible here is bacteria and fungi and the microbial degradation of organic material that goes in. And we can use DNA methods now to see which bacteria and fungi are important in that process. We have nutrient cycling, and here we have more targeted organisms depending upon the nutrients that we're thinking about. If it's nitrogen, it might be denitrifying bacteria. Mycorrhiza, we might think about phosphorus. We also have the role of nematodes in terms of grazing upon the bacteria and the fungi and therefore further releasing nutrients within the system. And we can measure that in a range of different methods. We can use functional genes methodologies, for example, to measure what specific types of bacteria are present and how they respond in a particular environment. We can look at the abundance of mycorrhizal fungi or the presence of arbuscular mycorrhizal fungi. For the nematodes, we can extract and identify them. Earthworms are critical for the water regulation and purification through the processes of bioturbation and aggregation, which I mentioned to you. And finally, for disease management, actually, diversity itself is the critical part here. So having a bit of everything. But again, I talked about we can use, really study the role of bacteria and fungi in terms of resistance and defense support to plant health. And we can use DNA-based methods to look at that. And there are many, many papers in the literature that support that. So my final slide really talks about, well, how do we then know what to measure? And this is a really interesting question, because for many, many years, we've had monitoring systems on our soils across the world. And we're very focused, and also in the questions that came today, about the chemical processes that take place within our soils, perhaps because they're easier to measure. Perhaps sampling the soils and running it in a laboratory is better. But what happens is, in the graph that you see here by Bunneman et al., which was published in 2018, and it's a very good paper, I would highly recommend it, they show the frequency of a soil quality indicator. So how often it's been applied within a monitoring or a measuring system. And of course, organic carbon and pH and phosphorus are the most commonly applied, along with nitrogen, electrical conductivity, and cation exchange capacity. These are the most regularly applied indicators of soil quality. There's also some soil physical indicators that are indicated in the blue on the graph. And these can include things like the water storage capacity, the bulk density, the texture of a system. And these are frequently collected, but actually once you start talking about structural stability or the hydraulic conductivity, so the movement of water through the soil or the rate at which the water flows through the soil. These are quite difficult measurements and so are infrequently applied. And the biological methods are indicated in the green bars. And what you will see is that there's very few studies where biological measures are applied on a frequent basis. And we need to really think about why that is. Because actually, soil biology is essential for all soil functions that take place in the soil. They're the drivers of all these processes. They need the chemical and the physical environment, but they actually drive the processes themselves. They're also very sensitive to the soil disturbances. So they're often a good early warning system when we're monitoring these soils. And they can often be linked to the chemical and physical processes. So we can understand the interaction that they have with the chemical and physical conditions. And yet we very rarely find them applied commonly within monitoring systems. So I think actually moving forward as scientists within this environment, we really have to emphasize the importance of monitoring the biological component of the soil and how we can do that in an effective and standardized and harmonized way across the globe in order to really understand the importance that these soil biomes play in providing us with sustainable plant and food production. Thank you very much. That's the end of my lecture for today, and I welcome further questions.
Thank you very much. Giving applause to Prof. Rachel then. So yeah, quite impressive presentations. So close by the issue on how frequent the soil biology become soil quality indicator. It means that the soil biology issue is still left behind on monitoring for sustainable plant growth. So it's actually also giving a bigger window for all scientists. Starting from now, we need to focus on developing indicator of soil quality using biological issue, sort of nitrogen mineralization, soil microbial biomass, measuring also soil respirations. It's actually quite simple. There's also several quite simple methods for identifying soil microbial biomass using fumigation method, soil respiration using potassium hydroxide trap. It's also quite cheap, I think. Yeah, Rachel. So, but it's still left behind so far.
Yeah.
So we can highlight that at least there are 5 topics of the sustainable development goal, which is influenced by the soil actions. So it means that actually the goal of the soil management actually want to sustain the plant growth, and it will help by maintaining soil diversity.
Yeah.
And the issue on soil biodiversity, it is because they have rules on maintaining nutrient cycling, also reducing the greenhouse gas emission impact, and also some issue on environment. So let's continue with the remaining questions. I would like to read some of it, and I'd like to know what is Rachel's perception. So how far actually balancing on soil biology properties, physical chemistry should be intercalated each other, and then in order to maintaining the level of soil quality and soil fertility. So it means that from this graph, it's actually clearly that we only focusing on soil chemistry, is that right?
Yeah, yeah, the red.
And then following by soil physical properties, and then the last one is soil biological. So how actually this graph should be in balance? So what's your opinion about this? It should be balanced or just left as it is?
I think traditionally, when we talk about soil quality, we have thought about methods and measurements that can be applied relatively easily within the soil, and that give us, I guess, a good basis for understanding some of the key properties that are taking place in the soil. For example, soil texture is critical. You need soil texture in order to be able to understand the rates of processes that are taking place, because the texture in itself provides the whole physical environment and can change the rate of different processes in the soil. However, we've always looked at it from a soil property perspective, so a measurement and property perspective. And I actually think that now we need to change how we look at that, and we need to think about the soil functions and the soil processes that we're interested in. And from that, we then define which are the best indicators, rather than just looking at a list of properties and saying which are the easiest to measure and which are the most reliable in their results. We need to think about which are the key processes that contribute to the functionality. And I think that will change how we monitor our soils into the future.
Okay, yes. So that's the conclusion. We're not only just focusing the role of each soil quality, but we have to look at on detail what is actually the process on and the functionality of the soil quality indicator itself.
Yeah, and I think then you would have a balance of biological, chemical, and physical.
Okay.
Property.
Yeah. So need to balance between all those soil indicators. So I think there's still one more questions, which is asking about how to recreate the or improving soil biodiversity, which is, for example, at the moment, when the soils did not has so many soil biodiversity at the moment, and then how actually recreate the environment, which part of the soil need to be recreated, which is coming from the physical or biological aspect. So what do you think?
That's a very good question. And it's not an easy to answer question, I have to say. I always say that in order to be able to answer a question like that, I need to see the place and the soil and the environment, the conditions that the soil is placed within. So hopefully one day I'll get to come to Indonesia and I can look at these soils with you and I can answer this question a little better. But I think the critical thing, obviously, is a good soil structure. I think
good soil structure. Okay.
A good soil structure really makes sure that you have good porosity in the soil, so good movement of water, and also good aeration in the soil, which is critical for the organisms that live in the soil. And in order to get a good soil structure, you also need then organic matter in the soil. So I think a prioritization on structure and organic matter really helps to enhance the habitat for these organisms to live within. And actually, you may feel that there's not many organisms living in the soil, but I think there's probably far more than you realize. It's just we can't always see them. And I think by ensuring a good soil structure and the incorporation of organic materials, always having plant cover is important. Where you have no plant cover, you often get degradation. That provides the conditions for the organisms to re-establish and to sort of fully develop. And so they're kind of the basics that I can give you, but obviously every situation is slightly different and it depends on the management of the system. So we really have to think very carefully how we manage our agricultural systems. And of course, things like contamination or too much repetitive tillage. Some tillage is good if it's done in the right way, but repetitive tillage at a depth on a very frequent basis, done in the wrong conditions, can be extremely degrading of the soil. So it's really bringing into the combination of the management of the system for the climatic conditions that system is within, to ensure good soil structure and carbon in the soil.
Okay, so that's I think the last questions that we have so far, but the time is just running out. Sorry for that.
No.
We're hoping that this is not the last. This is just the beginning of the future collaboration, not just sharing the experience through the seminars, but also for research. We are really hope that by your help, we could develop the soil biological indicator as one of the promising tools.
Mm-hmm.
So previously, I just say the soil biology is left behind, but for sure this is not.
Yeah.
This is not left behind, but we have a bigger opportunity for developing this indicator in the future for supporting and sustainable plant growth as one of the target of final global sustainable development goals. So Rachel, thank you very much for your speech, and we are hoping we will meet you again in the near future.
Thank you very much.
Thanks for your time. Thanks for the attention from the students, all the scientists who attending this seminars. I'm Cahyo Prayogo as the moderator for this seminar. Thank you very much for your attention and contributions. And we will see you in the next time. So thank you very much. Rachel, giving applause to Professor Rachel. And we'll take a picture together. Thank you so much.
Okay.
Wassalamualaikum warahmatullahi wabarakatuh. See you next time.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you, Professor.
Warahmatullahi wabarakatuh.
Selamat sore.
Thank you, ma'am.
Very much, ma'am.
Thank you,
Thank you,
ma'am.
Ma'am.
Mr. Rachel.
Thank you very much.
Hello to Professor.
Hati-hati.
See you.
ma'am.
Thank you, ma'am. Very nice to meet you, ma'am.
Nice to meet you.